Okay, so now that you are familiar with mitosis, we can move on to meiosis. So also a lot of people call it meiosis, which is also a correct pronunciation. I call it meiosis because it reminds me uh, how to keep mitosis and meiosis straight, because meiosis, my mom and dad had to have sex to get me. So meiosis is going to be about making the sex cells. And the sex cells, if you remember from the previous video, are called gametes. And a gamete is either an egg or a sperm, just depending on if you're male or female. Now, meiosis, it starts with a germ cell. And a germ cell is already 2N. So that means it has two sets of chromosomes, one set from mom and one set from dad. And so 2N is going to be called diploid. So the di in diploid means two. So you can remember that. And it ends with 4 unique gametes, and I'm going to circle 4 unique gametes because in mitosis we ended up with 2 identical cells because the one cell divided, it multiplied inside its chromosomes inside exactly identically, and then it divided into 2 identical cells. In this case, in meiosis, we're going to end up with 4, not 2, and they're going to be all unique. Each one is of the 4 is going to be a different combination of chromosomes in the gamete. And each gamete is going to be just plain old N, not 2N, just plain old N, because it only has one set of chromosomes, which is called haploid. Now, if you don't remember what a germ cell is, because this whole thing in meiosis starts with a germ cell, I'm going to play back just a few seconds of the original introductory video that describes germ cells real quick.
Instead of differentiation, the cells that are going to become gametes, which are the sex cells, the sperm and the egg, they turn into germ cells. They don't turn into liver cells, they don't turn into hair cells, they don't turn into skin cells. They turn into what's called germ cells, which are also diploid because
So
they
I hope
still
that
have
refreshed your memory
two
on what a
sets
germ cell
of
is.
23
So
chromosomes in there.
in
One set from
meiosis,
mom, one set
we're
from
going to
dad.
start with
So they're
this germ
germ
cell
cells
with
right
one
now.
germ cell, and
But
as we already said,
then once
that's
they reach
2N
germ
because
cells,
it has
they
two
go
sets of
through
chromosomes, one set from
meiosis.
mom
Now,
and one set from dad.
my mom and
My
dad have to
mom
have sex to make
and one from
me.
dad.
So
And
meiosis,
then
that's the sex
in the
cells,
first part of
they go through
meiosis, called
meiosis
meiosis
to become
1,
a
and I
sperm
wrote
or an
the
egg.
regular number 1 here, but usually it's written um, in Roman numerals so you can tell the difference. So it would be meiosis and then a capital I afterwards, which is just a Roman numeral for 1. So this is how it's actually written. So in this first half of meiosis, meiosis 1, we're going to start with this germ cell, and this germ cell is going to go through meiosis 1, and we're going to get, as a result, 2 haploid cells. Notice there's no 2 in here, it's just a plain old N. So when we go through meiosis 1, this germ cell up here which has two sets of chromosomes, when it creates these two uh, N cells, haploid cells, that's going to split this two sets of chromosomes, and one set of chromosomes is going to go into one cell, and another set of chromosomes is going to go into another cell. But that doesn't mean that this uh, one cell is mom and one cell is dad chromosomes. That's not what that means, because the chromosomes float around in the cell randomly and bump into each other and turn around and everything. So when you divide these chromosomes, these two sets of chromosomes, into their individual sets, one for each of these new cells, they're going to be all jumbled up and randomly mixed up together. So that's going to give us a good variety of genes, some from the mom, some from the dad. And that's why babies aren't going to look exactly like their mom or exactly like their dad. They're going to be like a combination of their mom and their dad. Then in meiosis 2, which is also technically written with a Roman numeral 2 after it, which would be a double I, meiosis 2. That's going to start with these two haploid cells that we made, and we're going to end up with four unique sex cells, the gametes. So these are going to be four. They're each going to have a different combination of chromosomes in there, and they are either going to be sperm or eggs, just depending on if we're talking about cells, sex cells of a male or a female. So, as you could probably guess from what I just went over, meiosis, it's going to be pretty much the same steps as mitosis, except you're going to go through each phase twice. Because remember, I said meiosis 1, the first part of meiosis, and meiosis 2, the second part of meiosis. So we're going to uh, have the germ cell is going to be hanging out in interphase, and it's going to go through all of the interphase phases. It's going to go through the growth phase. It's going to go through the replication phase, where, and this is how it gets a double set of chromosomes in there, because you already have a complete set of chromosomes in the germ cell. You already have two sets of chromosomes in the germ cell. So when it replicates, you're actually going to have four sets of chromosomes in the germ cell. And then it's going to go through uh, growth phase 2 and interphase. And then we're going to go over to meiosis 1, and that's where you go through prophase 1, metaphase 1, anaphase 1, and telophase 1. And be sure when you're taking the test and doing your assignments, if you're talking about meiosis, not mitosis, if you're talking about meiosis, you have to put what number it is at the end of each phase. Like mitosis, you would just write prophase. And if you just say prophase, everybody knows that you're talking about mitosis. So for meiosis, you have to specify, you have to say prophase 1 or prophase 2, or for any of the other phases, metaphase 1, metaphase 2, anaphase 1, anaphase 2, telophase 1, or telophase 2. So the cell is going to start out hanging out in interphase. It's going to go through all the interphase steps, and then it's going to go straight into meiosis 1. It's going to go through all of the steps that we went over in mitosis, and then it's going to go straight into meiosis 2, where it repeats those same steps. But there are just slight differences because we're dealing with a different number of chromosome sets in these sex cell for these or what are going to become sex cells. So pay attention to the slight differences. They're not too much. If you can follow along and get the gist of what I'm saying, it'll enable you to understand, um, just logically figure out what the differences are. There aren't too many. So we start out with a germ cell, and as you can see in this germ cell, these chromosomes are not in chromosome form. They're in chromatin. They're in chromatin or chromatid form called chromatin. So we don't call these chromosomes when they're in this unraveled uh, mess of segments here. We call them chromatin. And so since these chromosomes are actually chromatin, they're in chromatid form in this germ cell. What phase do you think I have drawn this germ cell in? So if you said interphase, then you are correct. And why do we know that this germ cell that I've drawn up here is in interphase? That's because the chromosomes are in chromatin form. They're not in chromosome form, they're in chromatid form. They're chromatin, which is this unwound, gloppy mess of strings all balled up together, uh, real, real messy, unwound. And if you don't remember this concept, go back to the first couple of videos when I'm talking about chromatin in the interphase stage and refresh your memory. But so if you said interphase and you knew that this germ cell is drawn in the interphase stage because these are chromatin, not chromosomes, then you are correct and we are ready to move on. Now, this chromatin that I drew of this germ cell, which as we went over, the germ cell is 2N. That means it has two sets of chromosomes, and there's one set of 23 from mom and one set of 23 from dad. So just to be clear, when I drew this chromatin in this cell, I made the purple uh, the 23 chromosomes from mom, and I made the blue the 23 chromosomes from dad. Don't count them, they're not actually 23. I just drew a little mess of string in here. And here is when they get to the meiosis, and this chromatin winds up into short little segments of sister chromatids. This is what they're going to look like. So we're going to have one chromosome from mom wound up and thickened like this in this little segment, and one chromosome from dad wound up and thickened, and they're going to be joined at the centromere, which is why they make a little X. It looks like a little X. Now, as I was saying, this germ cell that I drew is in interphase right now because you can tell that this is chromatin, not chromosomes. And the interphase, it's the same thing as when we went over mitosis, which is why I really wanted to make sure that you were comfortable with mitosis, because a lot of this is just going to be the same thing as mitosis, just in a different context. So the interphase cycle, same thing as mitosis. You start right after the cell division, and you have a new cell, and you go into the first growth phase. Then you go into the replication phase, which is called the S phase. And then you go into the second growth phase. And then after that, you get to this part in the cell cycle in the life of the cell where it is time to make new cells. And so that's when it goes into the meiosis stage. Now, what to pay attention to right before the meiosis stage, in this interphase, we're dealing with a germ cell, as I said. And a germ cell already has two sets of chromosomes in it. One set from mom, one set from dad. So it's already 2N. So if the germ cell is already 2N, but then we get down here to the replication phase, and that's where all of the chromatin duplicates itself, what's going to happen? Well, technically, the 2N with the two sets of chromosomes is going to become 4N for just a short time because it's going to have four sets of chromosomes because each of these two sets of chromosomes are going to duplicate themselves. So this 4, you're going to have two identical sets from mom and two identical sets from dad, starting at the S phase in the interphase right before meiosis 1. So as I just said, this S phase is the replication phase, and so the two sets of chromosomes duplicate while still in their chromatin form.